Welcome, everyone. My name is Sarah Meyer, and I am a member of the HEAL Data Stewardship Group. Thank you for joining today's Fresh Fair webinar titled Discovering New Science Through Data Reuse. Next slide, please. Today's webinar is being recorded and will be shared with all registrants. If at any point you have a question, please feel free to type it into the Q&A box at the bottom of your screen, and we will bring it up during our question and answer session after the presentations. On our next slide, you will see the objectives. Today's session will begin with an overview of how data reuse is supported within our HEAL data ecosystem. We will hear a real-world perspective on data reuse from our guest speaker, learn about key tools that enable cross-study discovery and reuse, and explore actionable next steps to identify reuse opportunities within HEAL. Next slide, please. The HEAL data ecosystem is designed to enable and streamline data reuse. Next slide. As many of you know, the NIH Data Sharing and Management Policy established clear requirements around data and metadata sharing. However, within HEAL, we view these requirements as more than a compliance exercise. The required elements of a data management and sharing plan, or DMSP, such as structured metadata, well-documented data dictionaries, and repository submissions, create a foundation for interoperability. The HEAL data ecosystem was intentionally designed to leverage those required components and connect them across a coordinated set of tools. In other words, compliance artifacts become infrastructure. And when standards are applied consistently and tools communicate with one another, data becomes more discoverable, more interoperable, and ultimately more reusable. Two of the core tools that operationalize this vision are the HEAL data platform and the HEAL semantic search, which we will discuss. To support HEAL investigators and research teams, the HEAL data ecosystem was developed as a platform and a semantic search environment that leverages metadata to connect information across repositories and databases while aligning with NIH data sharing policies and requirements. Within the ecosystem, data are deposited into HEAL-compliant repositories selected to meet the needs of different data types and study designs. The HEAL data platform enables discovery and access across those repositories and supports data sharing practices that align with NIH expectations for transparency and reuse. The teams behind these tools are also available to support investigators in navigating HEAL data sharing process and requirements. Specifically, the HEAL data platform provides a centralized catalog of HEAL data studies, which allow you to navigate the study landscape, find where data are housed, and provide workspaces to interact with available data. The HEAL semantic search helps you to discover studies by meaning and research concepts and identifying questionnaires and variables. Many HEAL studies are actively ongoing and/or completing primary analyses. As a result, the volume of data sets currently available for secondary analysis will continue to grow over time. What we are seeing now is the early phase of reuse within our ecosystem. As more studies complete their data collection and deposit data into repositories, the opportunities for cross-study and secondary data analysis will expand significantly. To help ground this real-world experience, we are pleased to welcome a researcher who will share use cases of data reuse within his work. We would like to welcome Dr. Ravi Mathur, who is a statistical geneticist at RTI International, whose research has focused on integrating diverse data sets for novel molecular discoveries. Dr. Mathur has broad experience with a variety of molecular data types, various bioinformatic approaches, and cloud ecosystems. Since joining RTI in 2017, he has served as a task lead on several projects funded by the National Institutes of Health, where he has led genome-wide association analyses, meta-analysis, genomic structure equation modeling, data sharing ecosystems, and multi-omics projects. At this point, I will turn the presentation over to Dr. Mathur.
Thank you, Sarah. Good afternoon, everyone. Um, as Sarah mentioned, I'm walking through my data reuse journey with a couple research use cases. Next slide. So my kind of overall timeline in data reuse started back in my PhD research doing bioinformatics analyses within Drosophila model organisms for a lot of genetic studies and a lot of data reuse within Flybase for that database. And now it has evolved into human based research, which the two applications I'll be talking about today is a reusable genomic workflow for public controls applied to COPD gene and opioid addiction data integration across different data sets for advanced studies on opioid addiction. Next slide. Thank you. Going into the reusable genomic workflow. Next slide. So, sorry, the title got cut off. The background on here of we're matching population controls to case-only controls has been done in epidemiology for many years. We've previously developed unbiased population controls approach for a genome-wide association study as referenced by the papers here. But a big proponent of this type of approach is consistency in ancestry and overlap in the genotype variants. So we have a lot of population of cohorts and controls that have been genotyped on specific arrays, looking at the Affymetric smokescreen that is used by the NIDA Genetics Consortium and others that don't have that substantial overlap. So what we've proposed to use and what we've published on is utilizing whole genome sequencing as population controls to address that limitations on variant overlap. And what we are using for that whole genome controls is the NHLBI TopMed cohorts with a breadth of for population controls. Next slide. So the TopMed cohort is a wide cohort. There is various different phenotypes that have been collected. It has over 180,000 participants, multiple different phenotypes within sleep, blood, heart, lung, and other phenotypes. It is a very ancestry rich and diverse cohort with 60% of the population with non-European ancestry. You can get more information on the TopMed with that link right there. Next slide. So the GWAS data sets that were used in this in our application, one was COPD gene. COPD gene is the largest study to investigate chronic obstructive pulmonary disease, COPD. Over 10,000 samples, includes both African American and European ancestries. And what's unique with this is there is whole genome sequencing and SNP array data from exactly the same samples. So the whole genome sequencing data is available via the TopMed program with that PHS number in dbGaP, and the array data is also available in dbGaP with that PHS number. Secondly, we use the collaborative genetic study for nicotine dependence, COGEN. It was initiated back in 2001. That includes nicotine-dependent cases and non-dependent smoking controls from Detroit and St. Louis. So there's over 2,900 blood samples, and it's used the Human Omni 2.0 array for about 2.9 million SNPs. And again, it's available in dbGaP with that PHS number. Next slide. Lastly, we use the ECLIPSE, the evaluation of COPD longitudinally to identify predictive surrogates endpoints. It's a longitudinal observational study of over 2,000 COPD subjects, along with smoking and non-smoking controls. It includes SNP array data from the Illumina HapMap v3.0 genotyping array for 500,000 SNP coverage. And again, it's available on dbGaP. Next slide. So in integrating array and whole genome sequencing, we wanted to make sure that there's no genotyping technology effects, and we had to go through many pre-processing steps to test out this integration across technologies. So that integration plan was a result of multiple testing to minimize false positives and replicate true signal. Next slide. So this graphic here shows the result of that integration protocol, what we're calling genotyping array whole genome sequencing merge or GAW merge. So that protocol um, looks at matching key features for array genotyped and your public whole genome sequencing data. For example, in our case, looking at are they all smoking cases, matching ages, matching sex proportions, and that kind of thing. From the whole genome sequencing, we extract out the step 2 there is we extract out the SNPs from the whole genome sequencing that overlap with the SNPs in the array. We go through an extensive QC process for that data, go through a separate phasing technique for each of the technologies, imputation. We found this re-imputation for the top med data to be really important. And finally, an association test and filtering the results based on minor allele frequency and imputation quality. And with that, this is or we go to the next slide. Using that, we've gone through, we have these 3 separate testing mechanisms where the first one is kind of to make sure there's no technical false positives of testing out same samples across array and whole genome sequencing in both European and African American, and making sure there is no technical signal in there. Our test B shown here is controlling type 1 error that which include include that when we integrate across array and whole genome sequencing data from different cohorts, that if we design it that there is no signal into this system, that meta-analysis will show no signal and thus controlling our type 1 error. And finally, our test C is replicating known GWAS hits. We run 3 different GWAS using Cogen with the COPD cases, Eclipse with the COPD cohort, and then Cogen with an African American. And we did a meta-analysis to integrate all those results. Next slide. And mention of all of this work is funded by work within BioData Catalyst as a BioData Catalyst Fellow. And with the replication of known signal, we used the known signal from Hobbs et al. 2017 paper. Again, it's a meta-analysis with for the first 15 eigenvectors as covariates and sex as those 2 covariates. And again, it's a meta-analysis across European and African ancestries. So what we see here is a replication of the known signal of the TGF beta 2, FAM13A, HHIP, CHRNA3, and CRIP2A6. Next slide. So that concludes that application. Happy to take questions. I know that was quick, but to walk through opioid addiction application and is switching gears a little bit. Next slide. So opioid addiction, as many of you may know, has strong heritability, but there's few genetic variants associations that have been identified and independently replicated. So there's 8 genome-wide associations studies that have been published with a range of case numbers from 100 to over 10,000. And the largest cohort of them within a European ancestry identified the genome-wide significant association RS1799971, which is the A118G variant for the LPRM1 gene. It's been highly tested for opioid addiction, and it has been independently replicated. So we were looking at, can we improve this for other variants that could be um, looking at opioid addiction. Next slide. So what we have here is the GENOA consortium, which is the Genetics of Opioid Addiction Consortium. And our wave 1 included 15 different cohorts as listed in table 1 on the right. 9 of those 15 cohorts included European ancestry samples. Each of those 9 have over 100 cases and controls. Each cohort performed their own QC and analysis using a standardized protocol, imputing to 1,000 genomes, RV tests for variant association testing. And one thing within this consortium is it includes a variety of case definitions. So both DSM criteria, methadone treatment qualification, heavy frequency of use, and including both opioid exposed and unexposed controls. And as you see in the table to the right, these are cohorts across the world, both decode that's collected in Iceland, Bulgaria, and then various US-based populations. Many of these cohorts are available with the PHS that's listed in the table. Some of them are with collaborations with the collecting PIs of those studies that myself and collaborators have establish those collaborations. So across those 9 cohorts, we did a meta-analysis across the cohorts using METAL. Next slide. And in addition to the GENOA, we integrated across different GWASs that are available to the public. One is the Psychiatric Genetics Consortium, Substance Use Disorder, GWAS that was published in 2020 that I had some genome-wide hits. Secondly, the Million Veterans Program identified a OPRM1 variant. Next slide. The Partners Health, also published in 2020, identified 2 hits. And finally, our genome-wide association study of GENOA identified another variant here. Next slide. And sorry, if you go back, my bad. What you'll see in each of these 4 analyses is the case count does vary across them, from PGC being around 4,500, MVP or over 10,000, Partners Health at over 1,000, and our Genoa at over 7,000. And it does show the um, inconsistency in the hits that are shown. But what we wanted to do is how can we take these four powerful GWAS results and integrate across them? Next slide. So to do that integration, we use genomic structure equation modeling, which is able to integrate across GWAS summary statistics. And one advantage of this is there was some sample overlap across those summary statistics, and the GSEM technique does account for that overlap. And the other thing, so we did observe strong SNP-based heritability for both diagnostic-based and frequency of use-based case definitions. And lastly, as I mentioned previously, there are different case definitions and different control definitions across these cohorts. And what we found is there's high genetic correlation between these different diagnostic-based and frequency-based phenotypes with an R of 1.05. Those were also similarly high as shown in the table in the top right. So then we conducted a genomic structure equation modeling as shown in figure 1A from our paper. The GSEM was set up as to have a PG of just one factor there, a single factor GSEM with the four cohorts integrated there. And we had high model fit for our model. Next slide. And this shows the results of the GSEM with on the left being the Manhattan plot, where on the x-axis is a position in your genome, and the y-axis being the significance of the p-value on a log base 10 scale. So what you see here is, with this whole integrated approach, now we had a total N of over 400,000, a case size of over 20,000, where we're able to identify the hit on chromosome 6, which is within the OPRM1 gene, the genome-wide significant hit on chromosome 16, RS1333582, which is within the furin gene. And finally, there's a nominally significant hit on chromosome 9, which is PPD6C. So we see some signal that we are following up on across this integrated data set. Next slide. So to kind of fully follow up on this, one thing that we just published was, so now we have these genome-wide association hits. Now what? What do we do next? So we went into of looking at drug repurposing of if there's drugs that are available that can target these data sets. So we have a integrative approach framework that combines the GWAS results that I presented here. We also conducted a gene expression analysis on similar opioid use cohorts. And then we integrated this with drug databases to identify opioid use drug repurposing candidates. So the genetic and expression analysis can identify gene targets. We also conducted some network-based analysis to integrate the genetic and expression results. And finally, we can, with the drug repurposing, we can expand the set of resources related to pharmacotherapies. So the figure that's shown on the right is our query of drug repurposing databases. So we queried Pharos, Open Targets, TTD, Drug Bank. There's thousands across these. They're pretty easy accessible via APIs. And what we are looking at these resources to see specific drug targets. Next slide. And this shows the results from that drug repurposing analysis. This is a similar Manhattan plot to our GSEM results. This is showing at the gene level. So each plot in this Manhattan plot is a gene within the genome. So what's shown towards the center of the screen is the OPRM1 gene. The size of the circle is the number of candidate compounds identified that target that gene. So as you can see, OPRM1 has a high number of compounds. The signal on chromosome 9 shows has a couple of different drug targets, and finally the signal on chromosome 16. Next slide. So kind of to recap all of that and kind of lessons learned from that from the data reuse journey, I want to kind of emphasize I think knowledge of the complete data collection and processing protocol is really key on knowing what arrays are conducted for the GWAS analyses to what pre-processing is done, the whole genome sequencing of what alignments were done, and kind of since this was all collected with repositories, is that repositories should collect and expose all of that metadata in all data stages for efficient data reuse. And I know data access is often a contentious subject of kind of following consented protocols and having it secure. But then what is that process of gaining access to it, of how do you make it not too onerous on secondary users to gain access to the data, of what is the data access request process? For example, there's a dbGaP DAR process that I know there's many steps to it, but I think it's something that can be improved on. And then finally, a lot of the data sets that I looked at, it includes data across NIH ICs, across ecosystems, across repositories. So having that interoperability across these systems is key, and I think it's greatly improving across the NIH landscape. And finally, with the drug repurposing, providing APIs for accessible pulling of some data and metadata enables quick integration interoperability across resources. Next slide. Thank you. Happy to take any questions, comments during the question and answer. I will turn it back over to Sarah.
Thank you, Ravi, for sharing your data reuse experience with the BioData Catalyst project and opioid addiction genetic variants. Before we move into the Q&A, we want to briefly connect Ravi's experience to the data reuse support available through the HEAL ecosystem and preview how we will explore those tools in our more hands-on tutorial, which will be held next month. Next slide, please. As Ravi noted, data reuse is often limited by practical barriers, including identifying relevant data sets, understanding what variables are available, and knowing where data are stored. Researchers also face added burden of navigating multiple repositories, which can be time-intensive, alongside uncertainty about whether funding supports reuse activities. The HEAL data sources will support reuse. As you see on the next slide, we have sources that are structured support to support reuse by depositing data in a HEAL compliant repository, such as dbGaP, which is one of our HEAL compliant repositories, and provide consistent access and governance. Both open and controlled access pathways and repositories enable responsible data sharing, while standardized metadata, both at the study level metadata level and the variable level metadata level, make data sets more findable, comparable, and usable. Together, this framework allows researchers to integrate HEAL data with external data sets, including related opioid and pain research, to generate new insights. And the HEAL ecosystem tools can help. As you see on the next slide, which is going to look very, very similar to a slide we've covered before, the HEAL data ecosystem has tools that are set up to help with data reuse. And these tools were thought about from the beginning of the project, and resources and time and access to data is improving consistently, and these tools are becoming more valuable and more useful in finding secondary data. Specifically, the HEAL data platform provides a centralized catalog of HEAL studies and points users to repositories where studies are hosted. The HEAL semantic search adds another layer by supporting concept-based discovery across studies and helping researchers identify related data sets, variables, and measures even when different terms are used. Together, these tools are helping lower some of the friction in the data reuse process. And in May, we are going to spend time walking through them in a more hands-on tutorial. Next slide, please. HEAL funding opportunities are now hosted on grants.gov. To identify HEAL funding opportunities, please make sure that you add HEAL to the search term and then filter the agency down to the NIH. A recent webinar put on by the Purpose Network invited Michael Oshinsky, who is the director of NINDS, Office of Preclinical Pain Research, to speak about updates around NIH HEAL funding and key areas to watch in the coming year, specifically in the pain research area. We will be posting the link to that webinar. It's incredibly useful for early investigators and people who are looking at identifying and both open and forecasted opportunities for funding, particularly around pain research. The same process, however, could be applied to any substance use or opioid research that is also happening in HEAL. And on the next slide, we've identified some possible funding paths for data reuse. We want to highlight a few of the mechanisms that we know support these efforts. Specifically, the R03 and the R21 can be particularly well suited for data reuse projects. The R03 grant supports smaller self-contained studies, including secondary analysis of existing data sets, while the R21 is designed for more exploratory or innovative research questions that may emerge from available data. We also want to highlight there's a newer Common Fund opportunity that was recently released, and we will provide that as a link both here and in our follow-up email. This opportunity supports pilot projects that use existing data sets, including integrating data across multiple Common Fund programs, which aligns closely with the goals of data reuse. In addition, we've compiled a curated list of relevant funding opportunities for you, and these will be shared after the webinar so you can explore options that best align to your research goals. Next slide, please. We encourage you to explore HEAL data that is already available through the HEAL data ecosystem to better understand what resources exist and how they can support your research. Please join us for a hands-on data reuse tutorial on May 21st from 1 to 2:30 p.m. to learn practical strategies for working with these data. As you plan ahead, consider available funding opportunities that can support and sustain data reuse efforts. And now we will move on to our Q&A session. And we are joined by our Q&A by several members of our team. Ravi Mathur has agreed to stay on for our Q&A. And we are also bringing on Hina Shah and Julie Horvath, who are members of the HEAL Stewards team, and Dr. Shum and Dr. Brianna Larrick of the HEAL platform team. And if all of our panelists could please turn on their cameras, we're going to stop screen sharing now and begin the Q&A session. So question number one is we're going to start with Ravi, and then we will open it up to the rest of the HEAL team. What are the data stages that you would like specifically what are these data stages that you would like specifically addressed or acknowledged by repositories? And would the transition of the data stages be captured as provenance, or would it be interesting or difficult to apply fair data practices?
Yeah, that's a great question. I think the different data stages, I look at it as kind of different stages on the data collection journeys. For example, with whole genome sequencing, it goes everything from the raw sequencing files, the FASTQs, to the alignment files, to any joint calls, to summary statistics, and that whole kind of data lifecycle. And I do think the transitions between them, what's done, what's processed, should be captured as provenance, in my opinion. Kind of getting that, since that will affect downstream what kind of results will happen, what kind of integration occurs.
Do other panelists have experience or suggestions when working with data that may show up either as raw data or processed data or other stages where you may have transforming data throughout the research project and how that works in repository submissions? I do know with qualitative data in particular, you'll often see it labeled either as raw data or transcripts or different levels of variables or themes. And so often on the qualitative side of research, people will label their data sets or their artifacts based on the stage that the data was in as they went through it. And that's one way that we see it documented in a qualitative work, which is a bit different than it's often documented, such as raw or processed imaging with other types of data. So, okay. All right. Then we will move on to question two, which is, can you expand on your thoughts for what can be done to increase interoperability?
Yeah, I think that's a great question, and there's a lot of work across NIH happening in interoperability, to my knowledge. I think, and a lot of that, I will state that a lot of that interoperability work I'm not directly involved in, but kind of on the other side of the user side. But I would say one thing is standards can be different across repositories of how it's stored, how variables are stored, how you search things. So it's like from a user point of view, you're transitioning of how does dbGaP think about things, how does HEAL think about things, and if that's different. But so having it maybe a standard approach across NIH's ecosystems, or I mean that's above my pay grade, but that's what I was thinking of, kind of having a bit more standards across the board, that you have a little bit more homogeneous. And that is I'm not saying that's a big task, I would say, um, across the different involved fields. I'm sure the HEAL data stewards have other thoughts on it.
Phil, go ahead.
Yeah, well, I just the standards, that's key, for data packaging and for storage of metadata with them. I'm less sanguine about the idea that there would be one uniformly adopted standard, only because for one, it's challenging, and for two, some standards do vary slightly with important distinctions. And so I think what certainly can be done is to have tools and to make it much easier to translate between those standards. So in other words, being prepared to bring those together and to harmonize them automatically, as long as they follow a standard that's well documented, that's possible. And I think that's something that certainly could be done in the genetics genomics world, but also one of the things that we together as the Heal Data ecosystem have tried to make some progress in.
Any other comments for panelists before we move to the next question?
I guess the only other piece I would add in is the machine readable formats are going to be really important because I think we're trying to get a lot more automation set up and get things to more smoothly transition. So I think that's one of the things stewards are definitely working on to get the machine readable aspects so that there's less burden on some of the researchers for reuse.
And I guess I would add just to kind of bring the point Phil was making and Julie, like all the way home for users, for Heal researchers. One way we've been trying to realize this in the Heal data ecosystem is creation of the Heal variable level metadata schema and our standards around that. So when you're creating your dictionary and deciding which information to include about your data set in that file, that is our attempt in part to address this.
And that actually leads really well into the next question that we're going to ask, which is, given today's technological landscape, how do you think that data reuse and sharing should be approached? And I guess how have you seen it approached in the work that you are doing might be a more focused question to ask.
Well, I definitely think the machine readable formats and processing is one thing the stewards are really working toward, working with investigators to try and get them to make these data dictionaries or their variable level metadata more accessible to everyone and just make sure that the standardized formats are consistent. That's definitely one way. And we're using a lot of automated pipelines now to make it more seamless so that researchers have less upfront work creating some of these files and then making them more accessible to others. So that's definitely, technology is really improving, and we're hoping that some of these formats are going to be really accessible to everyone and make things more seamless. So there's less burden on the researchers and less burden on the people who need to reuse the data, because that's really one of the challenges is, I think Ravi was mentioning it was really important to have some of these data sets standardized and really make sure the variables are all consistent. And so sometimes they're just really small inconsistencies that make data reuse challenging. And so if we could get some of these set up and more streamlined, I think that'll make the process more smooth for everyone.
On that, I would add, in terms of data being approachable, particularly for those conducting human subject studies, in a lot of cases, for real valid reasons, the entirety of the data set is not able to be shared openly. It must be some sort of controlled access. There's a review process to apply to get access to the data. But if there is a subset of those data that can be shared only, that doesn't have identifiable information, but still provides value to the research community, it is also true that data that are openly available are used more often than those that have a controlled access requirement. So that's something else people can do to try to encourage use of parts of their data.
Mm-hmm.
I will add a little more to it. So everything that has been said is true, but I feel like having as much information about your data as part of the data package that you make available is super useful. Any kind of information, whatever protocols you have used, how they have been used, what kind of data you are collecting. If adding a data dictionary or anything that can describe your data set as much as possible. And that can be also made publicly available, as Brianna was mentioning, without sharing the data, so that anybody who's coming to look to reuse the data, the whole point is so that, people are not spending time and money to collect the data that you have, that you already have, but to make it reusable, they need to understand what is collected as well. So, and metadata plays a big role in that. And we are trying really hard as part of the Heal Data ecosystem to make all of that metadata, come together and give you a picture to the reuse, to reuse possible reuse folks. So as much information as possible on the data packages is always helpful.
Thank you for your insights. Our next question, what kinds of information are most helpful for researchers when they are deciding if a data set could support their research question? I think we'll start, Ravi. What was important with the data reuse work that you did, and then we'll move to the Heal Stewards and Heal platform for comments.
Yeah, I think for my work, it's kind of one of like for the public controls, it has to match. We did like a quick matching on like age and sex, those kind of demographics that are we're comparing apples to apples in the from the genetic sense. Um, I mean, it's of the other thing of what kind of data do they have?
Mm.
Right? Of is it whole genome sequencing? Is it multi-omics for different multi-omics of um, so kind of having that of both the clinical data and the omic data being searchable and apparent of what kind of data is there, what level of the data is there to be flexible for different reuse applications.
I do know our May tutorial, we're going to be giving an example of how data sets were chosen for a data reuse tutorial that we were doing in May. So we will have some practical kind of walk through pieces of how we choose data sets and decide on research questions in that tutorial. So there'll be more coming on that. But I don't know, Hina, or Julie, if you had any initial thoughts you wanted to share about deciding whether a data set could support a research question.
So to me, it really depends on what the research question is. But if it is something that is relevant to standardized variables, for example, you might want to look at I come back to metadata, but you might want to look at the metadata and see what the researchers or the data package has actually collected, what they have, what kind of research they have done, and if they have data dictionaries, do they seem to collect the variables that you are looking for? And I think Heal Semantic Search and Heal Data Platform both have ways of looking at the metadata and some of that information as well.
Yeah, ultimately, I think it's the kinds of information that the Heal data ecosystem is asking Heal investigators to provide. So when you look at your Heal investigator checklist, you're asked to register your study, pick a repository. But that other information we're collecting, the study level metadata, information about the type of study you're conducting, the types of data you're collecting, outcome measurements, primary goals of your research. That sort of information, along with the information in the variable level metadata, the data dictionary, information about the outcome measures and variables in your data set. As you're providing that for your own study, you can look at it through this lens of what do people need to understand the study that I conducted and the data files and associated files that I'm sharing now. So the things we're asking you for are really to facilitate the secondary analysis and are the things that you should look at when evaluating other data sets.
Mm-hmm.
And I would just add quickly there, as many of you may know, NIH has done a huge amount of effort to try and increase data quality for HEAL-funded data through the specification of common data elements for primarily for collection of data with pain research. And I think the questioner asked about filtering. You can go to the HEAL data platform and filter based on use of those CDEs. So quality is a complicated thing with lots of aspects, as I know you know, but that's one of them, and that's something that you can make use of immediately.
Mhm. And we've also been asked a similar question. Can data management and sharing plans developed for the HEAL initiatives or repository-specific data sharing plans, such as foundation grants or publication requirements, can they be leveraged to inform and streamline data sharing for reuse across the broader research ecosystem?
So if I may, Sarah, I have one quick response to that. There may be more. You'll hear more at the subsequent webinar that was announced about the workspaces, the online workspaces within the HEAL data platform. That's a FedRAMP moderate platform that's accessible to any researchers who want to use it. And so to speak directly to this question, yes. In principle, we work directly with each of the 24 or so HEAL compliant repositories. And so a data management sharing plan that essentially says, I'm going to work with these data inside of a HEAL data platform workspace should be transferable and pretty efficient to reuse across the different repositories. That, in fact, is one of the intentions of the way this was set up.
Are there any other thoughts before we move to the next question? All right. Switching gears a bit. I have a question about semantic search and HEAL. Even when relevant data is found, it can take a lot of time to determine whether it is fit for purpose or of sufficient quality. Does the HEAL platform or HEAL semantic search offer features to assess data quality during search or at runtime, such as allowing users to search or filter based on quality indicators? Hina, I think we'll start off with you.
Yeah, sure. This is a great question. And I'll like assessing the quality of a data package does take a little bit of um, digging and digging into the actual data package. And uh, we haven't, uh, while we haven't necessarily done it yet as part of the Heal Semantic Search development, we do have probably some ways of indicating metadata completeness. And I'm sure Heal Data Platform also has that information, with the assumption that hopefully if you can have enough information about the study, like if you have a good SLMD filled in, and if you have a good data dictionary or information about the data sets themselves, uh, you would assume that the data is also as complete as possible. We are trying in Heal Semantic Search to add more information about the data sets, but they're still part of our development process, and we will add certain indicators. One of the indicators could be CDE compliance with the data dictionary that has been submitted, but we understand that that's not the full story of all the HEAL studies. But yeah, we are thinking about it in real continue development. Brianna and Phil.
Yeah, on the platform side, this also is something that we've been thinking about quite a bit. And while we're doing what we can to facilitate generation of high quality data sets that are shareable, at this time, we don't have any ability to filter on the platform by any sort of quality index of the shared data. But yeah, this is definitely a topic of interest.
Mhm.
All right. So our next question, we're condensing a bit. So if this was your question, if there are other pieces that you would like us to answer, feel free to reply in chat if there's other pieces you would like us to address. But initially, the question is going to be, how does HEAL handle situations where a derived product, like a data atlas, meaning data from multiple data sets across different repositories, is published before its underlying data sets are publicly available, particularly regarding citation, access control, or coordinated data release? So I don't know if the HEAL stewards want to take this on first, and then Ravi, I don't know if you have examples of the difference of, have you ever published like process data before raw, or other cases where a more completely packaged but smaller product would be submitted before the larger full data set is available?
I can comment a little bit. So the drug repurposing example that I gave, there was a little bit of a coordinated effort of making sure the GWAS was published and available. The gene expression was published and available. And then that drug repurposing, they were kind of all moving a little bit in parallel. And like, so yes, there was a little bit of coordination of what's published and what's available and kind of, um, but we coordinated it to make sure that each part of the integration is published before the full integration is published. I haven't had a case where that wasn't the case.
Are there other comments on this question?
The one thing I would simply say is that the repositories that we work with, retain complete control over, data releasing and data governance. And so, um, we can add information, we can do those kinds of things, but ultimately it is the individual repositories together with their data creators or submitters who, who have to determine these kinds of timing issues.
Mhm.
So that leads us to the last question of our webinar today. What is a typical workflow that investigators can use with HEAL Semantic Search and the HEAL Data Platform to identify data sets that are most likely to be analysis ready for a secondary research project?
I can give a very brief overview. uh, but the way, at least in my vision, you would start off either doing a research landscape analysis, or how, if you have a particular research question in mind, come to Heal Semantic Search, see where the data is available, what are the common data elements, if you are using common data elements, and if there are any studies that use the same common data elements among different data sets. Try to figure out variables, shortlist some of the studies, and you can navigate those, bring those back to Heal Data Platform. There are search tools available for Heal Data Platform as well, and I'll let Brian and Phil speak to it. But the Heal Data Platform has a great way of analyzing your data sets, analyzing those data sets within their workspaces as well.
Yeah,
But a webinar. Sorry.
I missed the last thing you said, Hina.
No, I was going to say come to the May tutorial. We
Oh,
will
yeah.
definitely go through it in detail, but please go ahead. Sorry.
Yeah, the Heal Data platform also offers a lot of search and filtering tools to help investigators identify studies and data sets of interest. The normal free text search sorts of things. We have filtering that will help you narrow down the search results to studies that are in the population that you're looking for, or the specific data types that you're looking for, study types. You can also search through the variable level metadata that have been submitted. So if you're looking for data sets that contain a specific variable or a specific common data element or instrument, you can filter for that information as well. Once you've identified a study or set of studies that you'd like to look more closely at, if those studies have shared their variable level metadata, you can download those files, take a look at those. You can open files that have been shared in a Heal workspace. And then in the next webinar, we'll be providing some more hands-on demonstrations of that, executing that process. But ultimately, you can identify your one or more studies that you'd like to look into and then access those data through the Heal Data platform. And then just to be explicit about it, the Heal Data platform does not manage authorization for any shared data files. That rests solely in the repositories. So our goal is any data set that you have access to from the repository, you have that same access through the platform. So if it's a controlled access data set and you're not authorized at the repository, you won't be authorized to the Heal Data platform too. So that's how that works. Phil, is there anything you would add to that?
Nope, that was perfect.
All right.
Well, we would like to thank all of our presenters and members of our Q&A team today. The Stewards and the Heal Data Platform team have a variety of resources available, and we will provide links both in the chat and then in our follow-up email. Please connect with us at any time to ask any questions or to set up a consultation. We want to thank you for attending today's webinar. And in addition to the recording of the webinar, we will also email everyone who has requested our list of resources and links that are used during the webinar. The slide deck will also be shared, and we will include a link to our Slack channel and connect with us forms that have been located in the chat. We hope that you reach out to us with any questions and support that you need as you embark on your FAIR journey to maximize the value of Healed data. We look forward to seeing you at our next webinar tutorial, actually, which will be on Thursday, May 21st at 1 p.m. It will be a 90-minute interactive tutorial session around Heal tools for data reuse. In preparation for that tutorial, we would like you to familiarize yourself with the Heal Data platform and Heal semantic search. And our follow-up email from this webinar will include some links you could review. Thank you again and have a wonderful day.